I'm going to go pretty fast. When we think about probability, you know, there's always some outcome that we're interested in, and we want to figure out the chance of that outcome happening. And so here we're just using dice, you know, very simple. So if we have one die and the outcome is that we roll a 6, that's pretty easy. It's just 1/6, okay? Because there's 6 sides to the die. All right, now we have, let's imagine a scenario where we have 2 dice. And the question is, okay, what's the chance of rolling a 6 on both of those 2 die? Well, there's 2 things that can happen. So we get a 6 on the first die, and then we have to get a 6 on the second die. All right, chance of rolling a 6 on the first die is 1/6. Chance of rolling a 6 on the second die is also 1/6. And now, as it says down here, you know, it's important, we have a name for these. We call these independent events because, of course, the 2 die are totally independent of each other, you know. And in those cases where we have those, we have to combine those independent events, we're going to multiply them, right? And so it makes sense that this should be a very small number because we're saying, okay, these 2 events have to happen in a very specific way for us to get the outcome. So this is 1/36. I don't want to emphasize the word "and" up there, right? These are 2 things that both have to happen. And, you know, if you focus a little bit on the language, it can sometimes help you think through more complicated, when we're applying this in genetics, which can be a little more complicated. All right, now in the last example on this page, we have 1 die now, and we can either roll a 5 or a 6, right? So we have 2 ways of getting the outcome that we want. We'll accept a 5 or a 6. So again, we could write it this way. We could roll a 6, or we could roll a 5. Either one of those are going to make us happy. So the chance of rolling a 6 is going to be 1/6, right? And the chance of rolling a 5 is going to be also 1/6. But now, you know, if we're just rolling 1 die, we can't do both, right? We say these are mutually exclusive, right? We can get a 5 or we can get a 6, but we can't get both. And so in the case of these mutually exclusive chances, we're going to add those together. And so that gives us, you know, 2/6, or we take it all the way down to 1/3 if we reduce the fraction. So the and the or is what I want to bring your attention to, right? So if we're saying, if we use and when we're describing these things, we're saying, okay, this has to happen, and this has to happen, and this has to happen. And we're going to multiply those numbers together, and we expect a pretty small probability. When we have these mutually exclusive variables, then we're like, well, we could get a 5 or we could get a 6. And then our chances should be higher than any one of those 2 things happening individually. Because now there's 2 possible ways to get our outcome. All right. So that's just introducing, like, when you multiply, when you add, this idea of independent events and mutually exclusive events. Now we're going to start thinking about genetics here. And in this case, we'll just take a case where we have 2 parents who are both heterozygous for some recessive disorder or something like that. And we'll say they're going to have a kid. So the first outcome is, what is the chance that the child is going to be homozygous dominant? Now, I know you can, you know, you're thinking Punnett square, which is a good idea. You could always draw the Punnett square. And the Punnett square, by the way, it is a little probability calculator. You know, you're going to draw the Punnett square. You're going to have 4 boxes. They're all equal. They're all possible outcomes. And it gives you a really nice visual way of seeing how frequent each outcome is going to be. But let's do it using probability rules instead. So the chance of getting an A from the mother, if she's heterozygous, is 1/2. And you also have to get an A from the father. And with him being heterozygous, that's also 1/2. And so these are both 2 things that both have to happen to get the outcome. So we're going to multiply them. These are independent events. And that gives us a 1/4. But of course, you already know that because you could just picture the Punnett square in your head up here, and you know that, you know, 1/4 of all the possibilities are going to give you that genotype. Now, we can also ask this way: what's the chance that the child has a dominant phenotype? And we would write the genotype in this case this way. Now, there were some people that asked about this during the exam, and I realized maybe this wasn't clear to everybody. If we write this, we're saying, you know, that they have at least 1 dominant allele, and we're not sure what the other one is. So they are basically this, or they're homozygous dominant, or they're heterozygous. And we don't care which, because either of those, we're going to get the same dominant phenotype, right? So again, you can look at the Punnett square and you already know the answer, but we can figure it out using probability rules too. So with the parents being heterozygous, the chance of the child being homozygous dominant is 1/4. We just figured that out up here. Or the child could be heterozygous. And again, looking at our Punnett square up there, we see that that's half of the chances. And the child can only have one genotype. So this is definitely a case where these are mutually exclusive variables. It has to be one or the other. So those we're going to add together. And that gets us 3/4. And of course, we see, well, we already knew that, right? From looking at our Punnett square up there, we saw that 3 of the 4 boxes have those genotypes in them. So you're probably saying to yourself, okay, yeah, that's great. We can do this with probability, but why, what's the advantage? The Punnett square is a nice, easy thing to do. And that's definitely true at this point. And even if you're dealing with 1 gene or even 2 genes, the Punnett square, it's reasonable to do it with the Punnett square. However, we're also going to look at cases in the class here where we have maybe a phenotype that's determined by 3 or 4 or more genes. And for those, you can't draw the Punnett square. It would just get crazy to try and draw that out. And so instead we use probability rules. So that's why we're kind of introducing them here, and then we'll keep practicing them throughout the quarter.